Okay, quick review for population of evolutions. Remember that genetic variation is what makes evolution possible, and that comes from random mutations in the DNA. A population's gene pool is defined by its allele frequencies, meaning how many dominant and how many recessive alleles in the entire population. Hardy-Weinberg describes a non-evolving population, and remember, natural selection acts on individuals because individuals have random mutations that give adaptations that allow organisms to survive and have babies more effectively, but only populations evolve. So you have to know a species is a group of individuals interbreeding in nature for fertile offspring. Populations are localized groups of these same species, and a gene pool are all of the genes in a population at any given time. Reminder, diploid organisms all have 2 copies of every gene. The gene pool, you're just throwing all those into the pool. You need to know how to calculate allele frequencies. So for example, in this very small population of 3 flowers, how many big A's, or big dominant, so we have 1, 2, 3, over the total, which would be 6. For any population, you should be able to determine a genotype frequency, phenotype frequency, and allele frequencies. And you can look at that in any sort of population. Genotype frequencies can be tricky because you cannot differentiate very often between these 2 purple flowers, even though they have different genotypes. So in order to do that, you need to know the allele frequencies. By convention, dominant allele is P, recessive allele is Q. For any gene, P plus Q must equal 1. And you can calculate these, and I just want to show you that they're in my notes down here. We have how you do those calculations if you want to look back at those. Populations can change over time. Things can die. We can have new births. We can also have immigration, migration, changes like that. When allele frequencies change, that means really there has been microevolution. Microevolution really is the allele frequencies changing, meaning the population has evolved. And there's really nobody that doubts whether microevolution occurs or not. There are small changes in allele frequencies within populations all the time. There's no real dispute about that. Hardy-Weinberg Equilibrium describes if the population is not evolving, then allele frequencies must stay the same across generations. And there's 5 basic assumptions for that. No mutation. There must be random mating, completely random. There must be no gene flow, so nobody entering or leaving the population. There's got to be an infinite population size and no natural selection occurring. Right? So if these assumptions are met for a gene, the population if these assumptions are not met for a gene, the population may be evolving, meaning the allele frequencies may be changing. And if you look at this, the 5 assumptions here are really all mechanisms that drive evolution, right? So mutation, a change in the DNA which leads to a new version of a gene. So for example, in our little bugs here, we have a random mutation that changes an allele, and now you have a changing allele frequency. So mutations can lead to allele frequencies changing over time and therefore evolution. Non-random mating, aka selective mating. So if you have something such as assortative mating or disassortative mating, any sort of preference for mating can lead to changes in allele frequency and therefore evolution. Gene flow is another one. So if you have any, for example, migration or the movement of fertile organisms or their gametes, then you can get allele frequencies changing and evolution. Genetic drift. This is an example that is very drastic in small populations. Genetic drift is the chance event caused by unpredictable fluctuations in allele frequencies. For example, a hurricane, a tree falling, a wildfire, any kinds of those. So here, a chance event changed the population so that only these 3 beetles were around, the next generation then has very limited genetic diversity. So genetic drift can cause allele frequencies to change over time and therefore have the population evolve. Natural selection. Individuals better adapted for their environment reproduce at higher rates because of those traits. It's kind of what natural selection is. And for example, here we have dark bugs on a dark background versus light bugs on a dark background. The light ones are eaten by birds because they're more visible and less camouflaged, and therefore the next generation has changed allele frequencies. So again, natural selection can cause allele frequencies to change over time, meaning the population has evolved. The Hardy-Weinberg Equation predicts genotype frequencies if the population is in Hardy-Weinberg equilibrium, all right? Genotype frequencies. So here we have a small population of beetles, and you can calculate allele frequencies. Remember, we're just counting the number of big A's over the total, or the number of little a's over the total. This gives us our allele frequencies. The Hardy-Weinberg equation is looking at genotype frequencies. P squared is a genotype frequency. 2pq is the genotype frequency for a heterozygote. Q squared is the genotype frequency of homozygous recessive. And you can make predictions of those genotypes using this equation. Right? So if we know our allele frequencies, we can then predict what the genotype frequency is for homozygous dominant by taking p squared. So here's just another example of that, where we have our starting genotype frequencies, which we calculated here. We have 9%, 42%, and 49% based on these allele frequencies. Here are our genotype frequencies, and we put all of those into just a big pool of gametes, right? And the Hardy-Weinberg equilibrium, or the equation, is really based on this Punnett square, where we have p squared, 2pq, and q squared. So microevolution, again, is the change in allele frequencies in a population over generations, and there's 5 main causes: genetic mutation, selective mating, gene flow, gene drift, natural selection, right? Again, you a quick example of another thing called a bottleneck, where what happens is that the bottleneck severely reduces the population size and thereby genetic diversity. And this has happened in cheetah populations. So the population was very large. There was some bottleneck event thought to be maybe a disease that swept through the population. Population dropped a lot. If it drops too much, obviously organisms can become extinct, but many times bottlenecks can recover. However, the genetic diversity of that new population is far reduced. And the same thing happened with elephant seals in the 1800s. Another term that you should know is founder effect. This is when the mother population, this has to do with migration, when a very small subset of a larger population moves to a new location and only mates with themselves. So that reduces the genetic variation in a population, and that is when you can see a lot of genetic diseases come across, such as the Amish. Another example of the founder effect we can see with Ashkenazi Jewish population, which experienced obviously a bottleneck, but also a founder effect, where a lot of Jewish populations, a small portion, moved to a new location, and then they had a small genetic pool. Remember that when we're talking about natural selection and the survival of the fittest, we're really talking about not only survival, but the ability to reproduce, all right? So here we just have population of rabbits. Half of the white ones have been eaten, and now the next generation changes, right? You get a shift towards a different allele frequency. There's 3 kinds of ways that you can see shifts in phenotype distribution. This is called a stabilizing selection. Stabilizing selection is when the intermediate phenotypes have the higher, have the intermediate phenotype right here in the middle has the highest fitness, and therefore the curve narrows. This is called stabilizing selection. Examples, human birth weight. Another way that natural selection can shift phenotypes is called directional selection, when one extreme phenotype is the most fit. For example, the peppered moths after the Industrial Revolution. These dark moths were more fit. The last one is disruptive disruptive selection, a type of natural selection where both extremes are more fit than the intermediate, and you get this bimodal curve. Natural selection is not a mechanism to try to make the perfect organism, right? It acts on existing variations, right? So it acts on existing mutations that are already in the population. Natural selection operates only on the traits an organism already has. Adaptations are often compromises. So loud frogs attract mates, but they also attract predators. So there's always this kind of teeter-totter effect of what is advantageous, but also not compromising. Chance events are a major issue when we're talking about populations and how populations move. So chance events such as natural disasters, environments changing, those have drastic effects on allele frequencies, and we're seeing that with our changing population.